Hello students, in this module we'll be starting up with our new chapter of biology, and that is chapter 5, Principles of Inheritance and Variations. Now students, have you ever wondered why an elephant always gives birth only to a baby elephant and not some other animal? Or why a mango seed forms only a mango plant and not any other plant? Given that they do, are the offsprings identical to their parents? Or do they show difference in some of their characteristics? Have you ever wondered, students, why siblings sometimes look so similar to each other or sometimes even so different? Now, these and several related questions are dealt with scientifically in a branch of biology known as genetics. This subject deals with the inheritance as well as the variations of characters from parent to offsprings. Students, inheritance is the process by which characters are passed on from parent to progeny. It is the basis of heredity. Variation is the degree by which progeny differs from their parents. Now, talking about the Mendelian genetics, all living organisms, they reproduce, right? It results in the formation of offsprings of the same kind, and the resulting offspring most often do not totally resembles the parent. So you can see here the picture showing the families, they are resembling their grandparents, their parents. It is just because of this branch of studies that we are going to study in this chapter, students. So what exactly the term genetics is? See, it is the branch of life science that deals with the study of heredity and variations. Now, what is heredity? Heredity is the transmission of characters from parents to the offsprings, right? So whatever characters there are, they will be transmitting from parent to the offspring. This is what the heredity is. Now, talking about variation, student, it is the difference among the offsprings and with their parents. Now, what are the hereditary variations? These are genetical and inheritable. And environmental variations, these are acquired and non-heritable. Now, students, father of genetics, Gregor John Mendel, he did a lot of experiments and hybridization experiments with pea plant. He's regarded as the father of genetics. So now students will be studying about the Mendel's law. Fine. So he has given the first law, that was the law of inheritance. Now students, it was during the mid-19th century that headway was made in the understanding of inheritance, where Gregor Mendel conducted hybridization experiments on garden peas for 7 years and proposed the law of inheritance in living organisms. Now students, during Mendel's investigation into inheritance pattern, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. His experiments had a large sampling size which gave greater credibility to the data that he collected. Also, the confirmation of his inference from experiment on successive generation of his test plant. So you can see here, students, there were 7 pair of contrasting character. They are selected by Mendel for his experiment. Fine. So you can see here, these are the characters that were dominant and these are the characters that were recessive one. So the first one was height, where the dominant one is tall and the recessive trait is short. The second trait was seed shape. So the dominant one was round, fine, which is round in shape, and the recessive one is wrinkled. Similarly, talking about the seed color, the dominant one is yellow and the recessive one was green. Fine. And the seed coat, it was green as a dominant and recessive one was white. Similarly, talking about the pod shape and color, it was inflated and green in dominant one and constricted and yellow in case of recessive trait. Talking about the position of the flower, axial is the dominant one and terminal position is the recessive one. So these were the characters, the 7 pair of contrasting character that was selected by Mendel for his experiment, and he has taken the pea plant for that. So you can see here, students, he has done an experiment basically. But before that, we'll be studying about some of the terminologies in genetics which are very, very important, right? So now, the first one is factor, or now in today's, it is known as by the gene, fine? So we call it as gene. In Mendel's time, Mendel called this thing a factor. So functional unit of heredity responsible for the expression of character in the progeny, it is termed as gene. Now what is locus? The position of the gene on the chromosome, fine? Suppose it is a chromosome and there must be a position of the gene, fine? So this is termed as the locus. Now what is allele? The alternative form of the gene for a contrasting character present on identical locus of homologous chromosome. So that alternative form of a gene is said to be allele. Talking about the phenotype. So what is phenotype, students? The external appearance of an organism due to the influence of gene and environmental factors. Means the genetic constitution is not responsible here. But talking about genotype, the genetic constitution of an individual responsible for the phenotype. Now students, talking about the phenotypic ratio, it is basically the correct proportion of phenotype in population. And talking about the genotypic one, the correct proportion of genotype in population. Now we have two more terms, homozygous and heterozygous. Now homozygous are the individual having identical genes in an allelic pair for a character. For example, capital T and small t represents the homozygous conditions. And talking about the heterozygous, you can see here they are having unidentical genes in an allelic pair. For a character, example, we have capital T and small t. They are not identical. Fine. So they are heterozygous. Talking about the dominant gene, you must have studied these basic terms in your 10th standard as well, student. Now, the gene that expresses its character is in heterozygous condition is said to be dominant one, and which fails to express its character, it is said to be recessive. Now, what is hybrid? The progeny obtained by crossing two parents that differ in characters. Fine. And we have two types of cross, students. One is back cross and one is test cross. Now what is back cross? The cross between F1 hybrid and one of its parent. It is back cross. Fine. So F1 hybrid is basically the first filial generation that we've got from the fusion of male and female gamete. Fine. We have fused up with the fusion of male and female gamete results into this F1 hybrid. And if we are crossing this with one of its parent, it is said to be the back cross. Fine. And what is test cross? The cross between hybrid and its homozygous recessive parent. It is used to identify the genotype of the hybrid. Suppose we want to identify the genotype of any of the hybrid by its phenotypic characters. So it is done with the help of the test cross. Now students, Mendel selected pea plant because pure variety are available. Pea plants are easy to cultivate and life cycle of plants are only few months, so that results can be got early. Contrasting traits are observed. Flowers are bisexual and normally self-pollinated. Flowers can be cross-pollinated only manually and hybrids are fertile. So due to these several characteristics, Mendel selected this pea plant. Now there are 7 pair of contrasting character which he selected for his experiment, and these things we have already discussed. Now talking about the Mendelian law of heredity, students, law of paired factors. Fine. So what is law of paired factors? Factors are responsible for transmission of characters, and these are always present in pairs, which nowadays they are called genes. Now what is law of dominance? In hybrid, dominant character suppresses the expression of recessive character. Fine. So the one which is able to express itself is said to be the dominant character, and the one which is suppresses the expression, that is said to be recessive one. Now students, Mendel's first law or law of segregation of law of purity of gametes, it states that when a pair of factors for a character brought together in a hybrid, they segregate or separate during the formation of gametes. This is what he called law of segregation, which is also known as law of purity of gametes. Now talking about the inheritance of one gene, so this can be explained by Mendel's monohybrid cross. The cross between two parents differing in one pair of contrasting character is called monohybrid cross. So students, in monohybrid cross, we take the one pair of contrasting trait. So Mendel has also taken the one pair of contrasting trait in order to explain the inheritance of one gene by his monohybrid cross. So you can see here students, Mendel took one tall plant, fine, and one pure short, that is a dwarf plant. So these are the parent generation. So it is represented, always remember one thing, that alleles are in pairs, fine. So you can see here we have capital T, small t, because they should be in pair only. Now talking about their gametes, fine, they'll segregate during the gametes formation. That is what said in the law of segregation and purity of gametes. So you can see here this capital T is one of the gamete of this tall plant, and this small t is the gamete of this one, the dwarf one. So when they fuse together, the resultant F1 generation, or the first filial generation that we have got, was capital T and small t. Fine. Now students, in first generation, Mendel observed that all the plants were tall. Fine. So this recessive character was unable to express itself. It suppresses itself, and that is why the capital T represents the dominating character. Now he did the mating or the selfing. So the capital T and small t, when fused up with the capital T small t, that is the tall plant, fine, they were selfing was basically made between these two. So you can see the gametes here produced was capital T and small t, and similarly of this plant it was capital T and small t. So this is the Punnett square that has been made here. You can see on one side we have written the two gametes of one parent, and here similarly the two gametes of other parent. So what happens if we'll fuse this capital T with, if this capital T gets fused up with this capital T, the tall plant is formed. Similarly, if this T fused up with this one, this is formed. That is the capital T and small t. Fine. Similarly, here also you can see if capital T gets fused up with the small t, this thing is formed. And lastly, if small t gets fused up with the small t, this thing is formed. So you can see here students, talking about the F2 generation, we have the two types of ratio. Fine. Like the talking about the F1 generation, you can see the talking about their genotypic ratio, all were capital T small t. Fine. So it means we can see that phenotypically they all are tall. But talking about the F2 generation students, you can see here that their genotypes was 1:2:1. So what is genotypes? When genetic constituents are involved. So you can see here this capital T represents the pure tall plant, and this in the end you can see the small t students, this one is the pure dwarf. So you can see one is this and one is this, and middle you can see in between we have capital T small t and capital T small t. So these are in 2:1. So we can say 1:2:1. The ratio was this in their genotypic one. And talking about the phenotypic ratio students, phenotypic is what we can see physically. So we can see that 3 plants are tall and 1 is dwarf. So simply it means 3:1 ratio. Fine. I hope this monohybrid cross is clear to each one of you students. Simple hai bahut zyada. What exactly happened in parent generation? Basically Mendel has taken 2 plants. Fine. The one was completely like tall, the pure tall, and another one he has taken was this pure dwarf. So he has crossed them together. In F1 generation he has found all the plants were tall. But when he did the mating and selfing, so he's observed a lot of other characters. Fine. In F2 generation you can see the recessive one was also expressed, which is one of the character of the parent. Fine. So this was all about, you can see here in monohybrid cross. See, he has crossed this tall plant with the genotype capital T, and here dwarf, that is small t, small t. So they both are like this capital T was homozygous. Fine. They are homozygous. I have already explained you what are homozygous, and this trait is dominant. Why it is dominant, students? Because it was able to express itself. Fine. So you can see here when they, the parents, they both crossed together. There, so this capital T, small t was formed, and it it basically says this complete information says that all the plants, they were tall in F1 generation. But when they were self-pollinated, you can see the results, student. The genotypic ratio was 1:2:1, and the phenotypic was 3:1. Fine. Thank you, students.